muy prolífica, que ustedes van a ver posteriormente todo lo que él ha desarrollado. Pero primero quiero agradecerle a la OAICE, la Oficina de Cooperación Internacional de la Universidad de Costa Rica, por hacer posible que el doctor Stack esté por acá. Para nosotros es sumamente importante que personas de la talla del doctor Stack vengan, nos enseñen sus hallazgos, nos nutran, nos motiven, y eso también es posible gracias a el apoyo de entidades y de la misma universidad, del sistema de estudios de posgrado, la Facultad de Medicina, la Escuela de Medicina, el posgrado de Ciencias Biomédicas, el Centro de Investigación de Estructuras Microscópicas, que son los que también nos ayudan a que el curso surja. Entonces, bueno, primero le voy a dar la bienvenida al vicedecano del Sistema de Estudios de Posgrado, Pablo Bolaños, quien nos va eh, a dar unas palabras, y pues posteriormente les presento al doctor Stack ya con sus credenciales. Muchas gracias, doctor Pablo Bolaños, por acompañarnos acá y ser partícipe de la visita del doctor Stack.
Doctor, también muchas gracias. Este, chicos y chicas, todo lo que tiene que ver con oncología hoy en día se tiene que abordar de manera molecular. Entonces es muy importante estar al tanto de la literatura, saber lo que está ocurriendo. PD-1 y PD-L1 son súper importantes. Es un pathway eh, determinante a la hora de tratar todo lo que son eh, los tipos de cáncer más prevalentes. Entonces, el día de hoy lo que van a escuchar es como el state of the art de lo que está ocurriendo. Y la idea es eventualmente conectar eso al entorno costarricense y ver de qué manera podemos avanzar. Para los que no saben, el sistema de estudios de posgrado son todos los posgrados que tenemos en Costa Rica. Bueno, casi todos, la gran mayoría son de la UCR, son 250, que también incluye las especialidades médicas, ¿verdad? Este, Ustedes son prácticamente la crema innata de esta universidad en términos intelectuales, entonces es necesario que también consideren lo que va a ser su vida en posgrado, porque necesitamos gente que eventualmente haga doctorados, ¿verdad? Y que se eduque en las mejores universidades. El sistema de estudios de posgrado ayuda a que ustedes vayan al extranjero. Este, entonces es eso, es el ciclo, ¿verdad? De retroalimentación, ¿verdad? En talento que necesita la universidad. Y eso era lo primero que yo tenía que decir. Lo segundo, donde ustedes me ven el día de hoy acá, yo soy paciente de Charcot-Marie-Tooth. ¿Ustedes saben qué es eso? Es una neuropatía del sistema nervioso, pero es periférica. Yo no debería estar caminando el día de hoy. Yo no tengo mielina. No sé ni cómo estoy de pie. Esa es la magia del café. Y gracias a un docente del PPM en el 2015, se me diagnosticó y se me trató a tiempo. Entonces inicié un progreso de terapia y me tengo que tomar varias gabas al día, todos los días. Pero entonces el hecho de que esté sano y de que esté acá es gracias a que tenemos una comunidad médica UCR muy capaz que resuelve los problemas con antelación, ¿verdad? Entonces, tengan presente que ustedes como comunidad médica le van a salvar la tanda a un montón de gente como me la salvaron a mí, ¿verdad? Eso es, tienen ese poder. Voy a ser muy breve, ¿verdad? Lo primero es que estoy muy feliz de estar acá con esta charla, ¿verdad? Immuno-oncology Beyond PD-1, Emerging Therapeutic Strategies, del programa de posgrado en Ciencias Biomédicas. Muy feliz, ¿verdad? De toda esta comunidad, del doctor Fernando Morales. ¿El doctor está por acá? No puedo venir, ¿ok? El doctor Bujan Mosa, Bosa, tampoco puedo venir. Ok, el doctor John Stack está acá. Vos estás aquí. Bueno, y ustedes distinguida audiencia, ¿verdad? A todo el personal docente, administrativo, que se acerca a tener esta charla. Son discursos muy estructurados. El TEC tiene 50 años, ¿verdad? Tenemos todo lo que son las especialidades, que son 79, son un montón. ¿Verdad? De esas 57 especialidades, 26 son médicas, 15 quirúrgicas y 16 pediátricas, ¿verdad? Súper interesante. Todo lo que ustedes pueden hacer eventualmente con especialidades, tienen que saber hacia dónde van, cuál es su vocación, ¿verdad? Entonces, otra cosa aparte y modestia aparte, somos semilleros de investigación biomédica en este país. La investigación biomédica, ¿quién la hace en Costa Rica? Mayoritariamente la UCR ligada a la caja, ¿verdad? Este, bueno, ¿y qué sabemos nosotros de oncología y cáncer? Bueno, tenemos cifras para el primer mundo y para el mundo en desarrollo, ¿verdad? Son dos realidades distintas. En Costa Rica se diagnostican con cáncer más de 13,000 personas al año, 13,000 pacientes y 6,000 costarricenses, hombres y mujeres, mujeres, niños, adultos mayores, mueren por esta enfermedad. De hecho, se dice que nos acorta la esperanza de vida por lo menos en 2 años, el promedio, que es bastante, ¿verdad? Este, pero como la población está envejeciendo, para el 2040 ese número de pacientes diagnosticados podría doblarse, ¿verdad? Entonces ya de 13,000 pasaríamos a 26,000. Eso significa que nuestras estrategias, la forma en que abordamos la detección y el tratamiento tiene que cambiar, ¿verdad? Muy bien. Entonces, estamos buscando prevención, que es muy importante. El estilo de vida ayuda a prevenir, alivio o bienestar, sabiendo que hay aspectos genéticos que de igual forma te predisponen. No hay nada que puedas hacer. De hecho, se vuelve una cuestión casi que de frecuencias y de determinismo biológico. La idea es atraparlo cuando aparezca. Muy bien. ¿Qué sucede el día de hoy? Bueno, entendemos, ¿verdad? Que nos lo va a contar el doctor Stack, que PD-1 y una contraparte que es PD-L1 es súper importante, ¿verdad? En todo lo que tiene que ver con la desactivación de las células T, ¿verdad? Por parte de los tumores malignos. Ellos, o sea, se apaga la célula T y entonces ya el tumor escapa la detección. Llegamos a un punto de equilibrio donde empiezan a acumularse otra serie de mutaciones y eventualmente tenemos metástasis. Entonces la idea es desactivar a PD-1 cuando se pueda. ¿Y cómo lo vamos a desactivar? ¿Preguntas? Bueno, alguna gente dice que con dieta. Sí, la dieta keto. Bueno, alguna gente dice que eso se puede hacer. Está bien, ustedes piénsenlo, pero yo prefiero las vacunas con anticuerpos. Entonces, lo que vamos a hablar el día de hoy es qué tan avanzada está la investigación en PD-1 y PD-L1, ¿verdad? Y cómo entonces podemos trasladar un poquito de eso a Costa Rica, porque también tenemos que abordar lo que es el diagnóstico. Y bueno, ¿qué puedo decir? La ciencia es la búsqueda de la verdad, los posgrados esencialmente es buscar la verdad, buscar abordajes nuevos, buscar evidencia sobre cómo tratar el cáncer, ¿verdad? Y ojalá que podamos aprovechar el espacio del día de hoy para aprender un montón. Pregunten todo lo que puedan, ¿verdad? Ustedes tienen que preguntar todo lo que puedan porque esa es la única forma en que podemos avanzar como comunidad, ¿verdad? Y que tengan una excelente charla. Muchas gracias.
Muchas gracias al doctor Pablo Bolaños, vicedecano del CEP. Bueno, ahora sí les paso a presentar al doctor John Stack. Y pues les voy a hacer un resumen tremendamente breve porque son currículums muy impresionantes, muy largos. Pero bueno, el doctor Stack es full professor, que eso es el equivalente a catedrático en la Universidad de Montreal, en Canadá. Específicamente, él se desempeña como investigador y docente en la Facultad de Farmacia de la Universidad de Montreal y fundó y lidera actualmente desde el 2010 un laboratorio que estudia pues, los receptores adenosinérgicos y una parte interesante del metabolismo que es el metabolismo de los nucleótidos de adenina. El laboratorio del doctor Stack se llama el Purinergic Lab por purinas y ese laboratorio es parte del University of Montreal Hospital Research Center. El profesor es biólogo de formación y tiene pues su formación ya más de posgrado en inmunología del cáncer y acumula ya en su haber a joven edad más de 100 publicaciones, entre las que uno puede destacar publicaciones de Nature Reviews, Clinical Oncology, Cell Reports, PNAS y algunas revistas de muy alto prestigio y perfil. Es conocido por haber, de hecho, en el mundo se conoce el doctor Stack por haber identificado una enzima que es productora de adenosina, que se llama CD77, CD73, que es un blanco terapéutico que actualmente está en fases clínicas 3, ensayos clínicos de fase 3, en como targets inmuno-oncológicos. El doctor Stack es cofundador y miembro de la junta del Scientific Advisory Board de dos empresas biotecnológicas, una se llama Surface Oncology y la otra Domain Therapeutics. Y también es miembro de la junta de una iniciativa que se llama Biocan RX, co-líder de Montreal Cancer Consortium, que lo que busca es acelerar la entrega de medicinas personalizadas de cáncer a los pacientes. Doctor, you're welcome. Really, we are really delighted to have you here. For us, it's a very fantastic experience to have people of such a profile as you. And really, we are really honored. I know you have plenty of business going on in Canada, and still you decided to come to here, even though it was San Valentin Day. Probably your wife is gonna kill me if she meets me at some point, but I'm really glad, and we are very delighted to have you here. Thank you, and we are delighted to listen to you.
Muchas gracias. Gracias por la invitación de visitar su hermoso país. I will give my talk in English, lo siento. So today I'm going to talk about different topics, but the main subject is immunotherapy of cancer. So the subjects we're going to talk about today, first of all, a very brief overview of our immune cells that are involved during cancer immunotherapy. Then we're going to go over the concept of immune checkpoint. Then we're going to talk about what is recognized on the tumor cells by our immune system. So those are the tumor antigens. Then we're going to expand a little bit our notions on what does it take to stimulate an anti-tumor immune response. Then we're going to talk about emerging therapies beyond PD-1, which include also cell therapies and very unique technologies such as bispecific antibodies. And finally, we're going to talk briefly about oncolytic viruses and the role of the microbiome. So we have a lot to talk about, right? So why immunotherapy? Because in a tumor, you often find immune cells. In some cases, the immune cells are more important than the tumor cells. There can be more immune cells than tumor cells. This is an example of a breast cancer. And in brown, you see the T cells in the breast cancer. So you see a lot of T cells. Sometimes these T cells can kill the tumor cells. That's what we see on the right. This is a video in the Petri dish in lab where the T cell comes and kills this tumor cell that becomes red when it's killed. So we know that T cells can kill tumor cells. Let's go back to the basic immunology. So what does it take for a T cells to be activated? It takes at minimum 3 signals. The first signal comes from the receptor, so the TCR receptor, that recognizes this red antigen presented by antigen-presenting cells, what we call APC. So these APCs present antigens, and these are recognized by the T cell receptor. That's the first signal. The second signal is through a co-receptor called CD28, which binds molecules such as B7 on the APC. And the third signal is the production of cytokines from the antigen-presenting cells. So together, the 3 signals activate the T cells. Do you remember where that occurs? That generally occurs in our lymph nodes. So the lymph in our system drains our tissues. And then in our lymph node, we have antigen presentation, which leads to T cell activation. Then the T cells circulate, and then when they encounter the same antigen on an infected cells, then it can kill that cell. Then we have B cells, B lymphocytes. So these B cells produce antibodies. Just a quick reminder of the structure of an antibody. So it's a 2-chain molecule, a light chain and a heavy chain that are linked with disulfide bridges. And like a Y, and it looks like a Y, at the extremities of the Y, you have the antigen binding sites. So 2 antigen binding sites for each antibody, but the 2 are the same. Okay, let's go back in time. Let's go back in 1890. In 1890, in New York, there was a surgeon called William Coley who made the observation that some of his patients with sarcoma that had at the same time an infection saw their tumor regress and disappear. So he postulated the hypothesis that maybe there was something in the bacteria or something in the body in response to the bacteria that could kill the tumor. So what he did was to extract a liquid out of infected organs. Disgusting. So he took out the liquid and then he injected that into the tumors of his patients. And some patients actually had a response. Some patients, for some, the tumors shrank. So that was the first step of immunotherapy. Later on, in the modern times of immunology, 2 brilliant scientists postulated the idea that maybe our immune system could be able to control against the occurrence of cancer. So that was the concept of immunosurveillance. But in 1960, that was just a theory. They could not prove it. At that time, another brilliant scientist out of New York, Lloyd Earl, from the discovery of William Coley, went a bit further. And instead of extracting a toxin from bacterial infected tissue, so in this days, Coley did not know what bacteria he was working with. Instead of that, Lloyd Earl decided to inject the Bacillus calmette-guérin, which is normally responsible for tuberculosis. So, but he inactivated that bacillus, and injected it to patient with bladder cancer. And even now today, this is a main treatment of bladder cancer today. So what happened is that after injection of the BCG, there's an immune response, and that leads to the disappearance of the cancer. And then in the 1980s, we started to identify proteins that were produced by our immune system that we call cytokines. And one of the cytokines that were identified was interferon alpha, and another one was interleukin-2. And a surgeon from Washington, NIH, it's called Steven Rosenberg, decided to inject patients with cancer with these cytokines, with the idea, again, that maybe our immune system could destroy cancer. And indeed, he saw some activity. And Dr. Rosenberg went even further. What he did in 1992 was to surgically remove a tumor and then isolate just the immune cells from that tumor, and then put those immune cells in a Petri dish, expand them manyfold, and reinfuse those cells to the patient. I'll show you what happened. So this is one of the first examples of cellular immunotherapy. So a 56-year-old male with metastatic renal cell cancer who received high-dose IL-2 in January 94. Patient underwent a complete regression of all disease and remained disease-free 20 years later. So this is the first example of a cytokine-based immunotherapy. From this cytokine-based immunotherapy, Dr. Rosenberg developed a cell therapy. So interleukin-2, it's like a fuel. It's a fuel for the T cell. It makes the T cell divide. So he said, well, maybe if we take out the tumor from a patient, and then in the lab, we expose the tumor to IL-2, and then we can expand those cells. Maybe we can reinfuse those cells to the patient. And indeed, he saw spectacular results. Okay, that was in 1992, 1994. So you can see here, okay, a melanoma disappearing after 12 days of infusion of the patient's own immune cells. But that proved the hypothesis that our immune system can regulate or can help us control tumor appearance, you know? So in the 1990s, early 2000, we started to have access to transgenic animals. So we started manipulating the genome of animals for research purpose. And one of the things that was able to be done was to delete immune component in mice, and then challenge these mice with carcinogen that would otherwise give cancer. So this is a famous experiment from 2001 from Bob Schreiber's lab. So basically here what we're looking at is the size of tumor in mice. Each bar represents one mouse. And on top, the experiment is done in mice that have T cells, and in bottom in mice that don't have T cells. And you can see that in mice that don't have T cells, a vast majority of tumor progress, whereas in mice that have T cell, a majority of tumors regress and disappear. So that was the first experimental evidence that our immune system can control against the appearance of cancer. In parallel, there was at the same time correlative analysis of patients with cancer that revealed that the more T cells in the tumor, the better survival was for the patient. So specifically, work from George Kukas, now at Lausanne in Switzerland, established that ovarian cancer patient with high level of T cells have a better survival than patient with no T cells in the tumor. The same time, 2003, just about the same time as the experimental evidence. Okay, so now that we've set the table, let's talk a bit more about immune checkpoints. So what are immune checkpoints? Immune checkpoints are receptors on T cells that block their function. At the moment, there's two main checkpoints, the receptor CTLA-4 and the receptor PD-1. If you look here on the left, you have a T cell that recognizes the antigen presented by the antigen-presenting cells. And I said there's a second signal required with the molecule CD80, CD86. So once the T cells gets activated, the problem is that they start expressing this receptor called CTLA-4. And CTLA-4 actually blocks the function of the T cell. So if you come in with a molecule that inhibits the interaction between CTLA-4 and CD80 or 86, that reactivates the T cells. And so the other pathway that is very important is PD-1, PD-L1. This mainly occurs in the tumor. So in tumor cells, as well as other cells, but also on tumor cells, you have a ligand that is called here PD-L1. And the PD-L1 ligand, when it interacts with the PD-1 receptor, it blocks the T cell function. So again, when you come in with an antibody that blocks this interaction, you can rescue T cell function and eliminate tumor cells. So the main discoveries of CTLA-4, so we need to recognize the work of our peers. So the discovery came really from work of Arlene Shark, Tak Mak, Peter Lindsay in about 1995, where they published the role of CTLA-4 as an important immunoregulatory molecule. Then a scientist called Jim Allison took that discovery to a next step and said, well, maybe if we block CTLA-4, we could have an anti-tumor effect. And so Jim and his group published that in 1997 and worked with pharmaceutical company to develop an antibody against CTLA-4 that was first injected in 2000. CTLA-4 antibody was then approved in the 2010 after this phase 3 clinical trial. PD-1 was a different story. So PD-1 was identified by Tasuku Honjo and Gordon Freeman on T cells. And then by a series of serendipitous accident, antibodies were developed against PD-1. And then these antibodies were developed into a drug, mainly by BMS and Merck, and then followed by Genentech Roche. And these were also approved in the time of about 2013, 2014. Okay, so this was a change of paradigm because up until 2011, we were treating cancer by hitting the tumor cells. We're hitting the tumor cells with chemo, we're hitting the tumor cell with radio, we're hitting the tumor cell with surgery, with targeted therapy. But now we're hitting on a different nail. We're hitting on the immune cells. Okay, so the change of paradigm led to a Nobel Prize in 2018 to Jim Allison and Tatsuko Honjo. At the moment, there are over 20 types of cancer that are treated with an anti-PD-1 or PD-L1 inhibitor. That includes melanoma, renal cell cancer, small cell lung cancer, non-small cell lung cancer, bladder, etc. These immunotherapies can be life-changing for patients. Okay, I'll give you the most striking example. It is for patients with metastatic melanoma. So in about 20 years ago, this was what the patient received. They received chemotherapy. And what we're looking at is the survival of patient. So the melanoma-specific survival. When the patient historically received chemo, most patients died within 2 years. 90% of patients died. In 2025, about 60%, so 55 to 60% of patients live a healthy life. About 1 in 2 patients live a healthy life. And not only do they, gain years of life, but some of them actually are cured of their cancer. But for the first time, we can cure cancer. So that's nice for melanoma, but unfortunately, it's not the case for every cancer. So if we look at this response rate, the overall response rate, which is the combination of patients that have at least 30% reduction in their cancer. So if you look at the overall response rate across different cancer, we see that on average, there's about 20% of patients that benefit from immunotherapy. So the cancers that benefit most are Hodgkin's lymphoma, melanoma. The cancers that benefit the least, breast cancer, for instance. And like I just showed you, unfortunately, the most common cancers do not really benefit from these treatments. So breast cancer, prostate cancer are the most frequent cancers. But also colorectal cancer, pancreatic cancer, ovarian cancer, which are quite deadly diseases. Unfortunately, they do not very much benefit from immunotherapy. Oh, my slide got distorted, but that's not the problem. A few words on the side effects, because of course, no treatment is perfect. There's always a risk of side effects. But thankfully, the side effects of immunotherapy with PD-1 inhibitors are very modest. So mainly rash, skin rash in 20% of patients. Some elevation of liver enzyme, ALT, for instance, but in low-grade toxicity. And diarrhea, again, mainly low-grade, below grade 4. So diarrhea in 20% of patients. Very few, but some patients develop severe pneumonitis and severe colitis. And exceptionally, some patients can die of these toxicities. Okay, now next topic. What do the T cells recognize on the tumor cells? So we've been taught that T cells recognize antigens. So what are the antigens that are being recognized? When we get an infection, it's easy because those antigens come from outside of our body. So our immune system, is educated to tolerate ourselves, but not to tolerate the foreign invasions. But what about our own body? How come our immune cells can recognize our own cancer cells? Well, mainly because that cancer is a consequence of mutations. So the more the mutation, the more antigens that can be recognized by the immune system. So on the right-hand side, you have cancers that respond really well to immunotherapy, melanoma, lung cancer, bladder cancer. And what you can appreciate, if you look at the spectrum of mutation, so on the y-axis, it's the number of mutation. So you can see that these cancers that benefit from immunotherapy have a higher mutation burden. And if you look a little bit more, the survival data, for instance, in melanoma, this is a nice study that dichotomized the cohort in patients that had more than 100 mutation and patients that had less than 100 mutation. Small number, but still significant difference in survival. So the immunotherapy works well in cancer with a lot of mutations. So those are melanoma, lung. So how come? Well, because mutation and indel genomic rearrangements change the nature of our proteins. So the protein is now different, and because it's different, our immune system can recognize it. So based with this observation, Okay, some scientists postulated that maybe cancers that have deficiencies in repairing the mutations. So maybe the cancers that have deficiencies in repairing the mutations, well, maybe they could benefit from immunotherapy. So there's a category of cancers that are called mismatch repair deficient. So these cancers lack enzymes that are responsible for repairing DNA. And because they lack these enzymes, they accumulate more mutation. And so the idea is that tumors unable to repair mutations are maybe more sensitive to immunotherapy. And that was discovered a few years ago, probably as well. But for immunotherapy, it's spectacular. It's really spectacular. Like, I'll show you data in a couple of minutes. But for instance, these tumors that are deficient in DNA mismatch repair, virtually 95% of patients have a complete response to the immunotherapy without even giving chemo. Okay, I talked about PD-1, CTLA-4. But to generate an immune response against the tumor, you need more than just a T cells. You need a village. It takes a village to raise a T cell. And that village, well, starts with the production of antigen, like I just mentioned. And these antigens need to be presented by the antigen-presenting cells. That usually occurs in the lymph node. And then the T cells traffic, and they need to re-enter the tumor. So unfortunately, sometimes the tumors, they have roadblocks, like fibrosis, that blocks the entrance of the T cells. And then once the T cells is in the tumor, it needs to kill the tumors. And that can be regulated by professional cells, such as T regulatory cells, but it can also be regulated by macrophage fibroblasts, which in the tumor environment block the function of those T cells. In addition to the T cells, you have other effector cells, such as B cells that can produce antibody and present antigens, and NK cells. So the immunity cycle, the tumor immunity cycle, offers other possibilities of treatment beyond PD-1 and CTLA-4. Okay, can we predict which patient would respond to these immunotherapy? And why is this important? Because I showed you in some cancer, there's only 10% of patients that benefit. And because of that, we don't give those immunotherapy to these patients. But if we identified a biomarker, a signature that could help us select the patient, maybe we can give now the immunotherapy to the right patient. So can we identify a marker that helps us identify these patients that respond? So we asked that question a couple of years ago, and we turned to big data analysis. And in a very short few minutes, I'm going to just summarize our observation. So what we did with our colleagues was to gather the sequence of 3,600 patients that were treated with immunotherapy from 12 different types of cancer. Okay? If anyone is interested to digging into our algorithm, it's on predictio.ca. So we sequence, or we use the sequence to see if there was a gene signature that could be predictive of response. So what we computed was the odds ratio, which is the risk of relapse or the risk of response. So the odds ratio for each gene within all the patients that we had. And then we looked at the top 100 gene, and we calculated the mean expression, the average expression of this top 100 gene, and whether that had an effect on the ability to predict response. So the effect size here is shown as a function of the number of genes that we use in our threshold. So you can see there's a plateau at 100. So meaning that even if we add 200 gene, there's not much benefit to 100. But lower than 100, we lose the effect size. So if we look here on the left-hand side, this is the list of our 100 gene. Some of them are associated with resistance. Some of them are associated with sensitivity to immunotherapy. And most importantly, this is our new signature. When we compare our signature to all the other existing signature out there, we see that it's much more efficient at predicting the response. So this is the log p-value, and this is the odds ratio. And when we compare this signature to the number of mutations, I told you before, a number of mutation is a predictor of response. So the TMB is the tumor mutation burden. This is indicative of a good response, but the ability to predict is in the order of 60%, whereas our signature has an ability to predict in the order of 75%. So we're continuing to analyze sequencing data. This was one example, but I wanted to expose you to what we're doing. And at the moment, we do a lot of bioinformatics. And if you're interested in that space, in that field, there's a lot of needs. There's people looking for bioinformaticians all the time. Okay. Now, let's talk about new targets. So I'm going to talk about another T-cell checkpoint, and then talk about regulatory T-cells, and then about our work around adenosine. So I talked about PD-1. There's another T-cell receptor called LAG3. LAG3 for lymphocyte activated gene 3. So LAG3, okay, is expressed on the activated T-cells, but also on the T regulatory cells. On the activated T-cells, it interacts with the MHC molecule to suppress the development of memory T-cells. So this is a LAG3 molecule on the T-cells, and it binds to the MHC that normally interacts with the T-cell receptor. And there's been an antibody that was tested in melanoma patient that showed that when you combine this antibody against LAG3 called relatlimab, this antibody in combination with anti-PD-1 enhances survival compared to PD-1 alone. And this is the data I was referring to earlier in MMR deficient colon cancer. So in these patients that have deficiency in repairing their DNA, when you give an immunotherapy, that is the combination of anti-PD-1 and anti-LAG3, you see virtually 100% response rate. 80% of the patients in this trial had a complete response, a complete disappearance of the tumor. So very spectacular results published in 2023. If you're familiar with immunology, you must be familiar with regulatory T cells. And you know from your immunology class that immune tolerance is dependent on regulatory T cells. So there's a lot of interest in cancer, in oncology, to study the role of regulatory T cell and to deplete the Tregs as a mean of therapy. So there's a lot of work at the moment, and this is just highlighting some data. So in mice, you can see that the tumor size in the control group grows. The tumor size, if you give an antibody against CCR8 that is unable to bind the Fc receptor, you have no depletion of the Treg. But if you have this CCR8 antibody that is able to deplete the Treg, well, then you have total disappearance of the tumor. So for those of you who did some immunology, you will know that the Fc portion of the antibody is required for depletion of cells. So that's why this antibody here that depletes is able to cure the mice from cancer. Okay, in the next few minutes, I'm going to talk about our own work. Okay, I know it's a long day and you're a bit tired, but this is the most important part of my talk because it's my own work. Right? Okay, see. Oh, you cannot hear me? Okay. Okay. Usually I speak too loud, so okay. Apologize. So my lab studies the role of ATP and adenosine in cancer. And why is that important? Because we know that ATP is a source of energy, but maybe you don't know that ATP is also an inflammatory molecule. When cells die, they release ATP, and that can activate different receptors on our immune cells, such as the P2X7 receptor, which triggers inflammation. When there's too much tissue damage, their cells upregulate a family of enzymes called ectonucleotidase that deplete ATP into adenosine. This adenosine then suppresses our immune cells by acting on two different receptors, A2A receptor and A2B receptor. The adenosine inhibits the T cells because when its receptor gets activated, it accumulates cyclic AMP in the cell, which activates protein kinase A, which then activates CSK kinase, which inhibits the LCK kinase of the TCR. Why is this important for cancer? Because in cancer, you often have an upregulation of the enzymes that produce adenosine. So mutations in RAS, RAF, EGFR, P53 will lead to the expression of these enzymes. And you also have upregulation in response to hypoxia, to inflammation, and to cell dedifferentiation, which often happens in cancer. So when I started my lab, my hypothesis was that blocking the enzymes that produce adenosine will enhance anti-tumor immunity. So we did proof of concept in mice by studying mice deficient in CD73. And what we found was that mice were protected against cancer. So this is an experiment where we inject a carcinogen to the mice, and we look at survival over time. And we see an increased survival in the CD73 negative mice. And on the right-hand side, we're showing that the consequence of knocking out CD73 is that you have an increased infiltration of lymphocytes in the tumor. So from these observations, we developed antibodies that block CD73 and tested whether these antibodies could be efficient in mice. So this is an experiment where we inject tumors. Every line represents one mouse. So this is the control. This is the group treated with an anti-PD-1. So you see some mouse have a delay in tumor growth and some mice have a rejection. Almost the same profile for blocking CD73. But then when you block both together, you have a very nice synergistic effect where virtually no tumors grow in these mice. So we were very excited. So we collaborated with different pharmaceutical companies and tested that idea in patients. So this is the most mature data that was released 2 years ago, almost 3 years ago now. So this is a randomized clinical trial in patients with lung cancer. So they were randomized to receive the standard of care, which is an anti-PD-L1, or a new treatment, which is PD-L1 plus CD73. And you can clearly appreciate that the combination increased the survival of these patients. So now there's a phase 3, as Warner mentioned, a phase 3 open in this indication, and we're hopefully we'll see some data in the next year. We're also very encouraged about data in pancreatic cancer patients. So this is an example of a study published this year where patients were treated with anti-CD73 with an anti-PD-1 plus chemotherapy. And the conclusion was that in the patients that had high levels of the CD73, so patient had improved survival when they were treated with the new treatment. And similar results were observed in another trial, but this time targeting the other enzyme called CD39. So this other nucleotidase also participate in the production of adenosine. And here they identify a biomarker, which is HLA-DQ. And in the biomarker positive population, the response rate was 46% compared to 8% in the low biomarker population. So again, very encouraging results because you might be aware pancreatic cancer is a deadly disease with no treatment available. And in pancreatic cancer, again, this other company, Arcus, is now opening a randomized phase 3 worldwide to test the CD73 inhibitor in pancreatic cancer. Okay, do you have any question? Let's interrupt a little bit. Do you want to ask a question right now around this, or you want to wait at the end? Okay. The other topic I want to talk about is, if you have questions, raise your hands and I'll just stop talking. So the other big type of immunotherapy at the moment are cell therapies. And that's what I want to talk about next. So cell therapy right now mainly consists of CAR T-cell therapy. So what are CAR T-cells? So CAR is for chimeric antigen receptor. So they're modified, gene-modified T-cells. So the method is as follows. So the patient comes in, and we take from the patient, we take blood cells. And from the blood cells, we modify the T-cells to express a new receptor. And this new receptor is a chimeric receptor, is able to bind to tumor cells. So in this case, it binds to the CD19 molecule on the tumor cells. Then we reinfuse these cells into the patient, and the patient gets cure of this disease. So the molecular structure of these receptors looks like this. So I told you about the structure of an antibody. So that's a typical antibody. So the chimeric antigen receptor, we take the variant region of the antibody, so this small part. So we take the variant region, and we fuse that variant region to the intracellular domain of a T-cell receptor. So the T-cell receptor here, instead of being a normal T-cell receptor, it's linked to this antibody portion that recognizes the tumor antigen. So when the lymphocyte recognizes the tumor cells, it can kill that tumor cell. So for leukemia, for acute lymphoblastic leukemia, it's spectacular, about 90% response rate, okay? There's about 9 different CAR T cells approved at the moment for essentially leukemia. And there's also one approved for multiple myeloma. But there's a lot of work being done in labs around the world to expand the possibilities. And one of the most recent discovery or landmark discovery was made at Stanford University by Crystal McCall. And she reported these findings in childhood brain cancer. So she generated a CAR T cells that was able to recognize an antigen on glioblastoma. And she infused those CAR T cells in the brain directly of the kids. And she looked at whether that had any benefit. And you can see 7 out of the 10 patients had a benefit from that treatment. And 1 had a complete response and is still free of cancer 4 years after treatment. So spectacular results for this very deadly disease, which is brain cancer. The problem that we have for CAR T is that it's very difficult to manufacture, to produce, okay? Because it's a patient autologous sample. So the patient comes in, we take the cells from the patient, then we need to engineer the cells. And that can be done in the hospital, but most often it's done in a pharmaceutical company. So you would send those cells away to the US, they get modified and they ship them back, and then you reinfuse. So it's very complicated and it's very expensive. But there's a lot of work and very exciting work around developing a product that could be frozen away in every hospital and used like any other drug, like a frozen off-the-shelf cellular therapy. But to generate this off-the-shelf cell therapy, we need to knock out the normal T cell. We need to knock out the MHC molecule, so there's no allograft rejection. And so there's lots of things we need to do, but it's work in progress. And I'm convinced within the next couple of years, we're going to see that. The other possibility that is currently being used for cell therapy is the infusion of tumor-infiltrating lymphocyte. If you were listening, I already mentioned this earlier. I told you that Steve Rosenberg was one of the pioneers of this technique. So the first time that was done, it was in 1994. And it took a long time, but now it's in some hospital, it's available. So the idea, again, is you take a tumor, and in culture, you expand the T cells, and you reinfuse those T cells to the patient. And so last year, the FDA granted approval of a pharmaceutical product for treatment of melanoma that is resistant to immune checkpoint inhibition. And this product is sold by Iovance. So basically, what happens is the patient comes in, gets the surgery, and then the surgical sample gets shipped to the company. The company generates the cell product and then resend the cell product. You can do that at the company, which is very expensive, but more and more people are doing it in hospitals. So the Dutch are very at the forefront of this, and Canada will soon be the next country, I hope. So basically, we don't need to go through the pharma. We can do it ourselves. So the Dutch in Amsterdam, they do it in their hospital. So they take the T cells, they expand, and then they reinfuse. And my good colleague in Montreal is doing the same at the moment. Okay, I only have 2-3 slides, and then I'm finished. But these, I think, are the most exciting because it's the new technology. It's like science fiction, right? But science fiction that will occur, that will be reality soon, I think. So let's talk about vaccines. I hope you're pro-vaccines, because if you're not, you can go to the US if you're not. So here we're talking about treatment vaccine, so after the diagnosis. You remember I told you that tumors accumulate mutations, and these mutations can be recognized by our T cells. So here the idea is to identify these mutations. So the pipeline looks like this. So the patient comes in, there's a sample that gets taken, and then the sample of the tumor is sequenced. So we sequence to identify the mutations. And then we use AI algorithm to predict which mutation is the most likely immunogenic. And over the last 10 years, these algorithms have improved a lot, and they're very efficient in identifying the most immunogenic mutation. And then we select like the top 20 mutations, and we make an RNA vaccine. So this is what Moderna, BioNTech is doing at the moment. And with spectacular results. So this is data from a bit old, I think from last year, but still very impressive. So these are melanoma patients that receive either standard of care, which is the anti-PD-1, or the standard of care plus the vaccine. And look at the improvement. I mean, that's very spectacular. And the reason why at the end of the curve, you know, it combines is because the follow-up here, not every patient has reached follow-up. So disregard this because not every patient has reached that endpoint. But look at the split of the curve at 60, 80 weeks. It's very spectacular. So this is going to be a treatment approved next year, I think. So beyond this cancer vaccine, there's also new antibodies that are being developed. So traditionally, an antibody, I showed you, it looks like this. But you can fuse the variable region of two antibodies to generate a new molecule called a BiTE, a bispecific T-cell engager. So you fuse the variable region of an anti-tumor antibody with the variable region of an anti-CD3 antibody. So what happens is that this BiTE molecule can bind to the CD3, which is part of the T-cell receptor. It binds that T-cell, and then it binds the tumor cell. And by bringing the two together, it activates the T-cells to kill the tumor cell. And these bispecific antibodies, there's about a dozen approved at the moment, and there's more coming. You can, in addition to activate T cells, you can activate NK cells, which is another immune cell population, which is very important. And so there's a lot of exciting research in that field. Finally, oncolytic viruses are also an emerging technology. There's one oncolytic virus approved for melanoma. So why oncolytic virus? Because we know that some virus can divide better in cancer cells than normal cells. For instance, vaccinia virus, if you're infected or if you inject yourself, your cells will destroy the virus. But the tumor cells have some defects, which makes them more susceptible to the virus. So when the patients receive this virus, the virus proliferates only in the tumor and destroys the tumor. So it's oncolytic. So there's one approved and there's more coming. You can also arm these oncolytic viruses to produce like a manufacture of other drugs. So the last topic is the microbiome, and then I'm done. So why the microbiome? So maybe you don't know, but your body is composed mainly of bacteria. Okay, you're not human. Okay? Nearly 3/4 of our body is composed of bacterial cells. The mass of our gut microbiome is as heavy as our brain. So we have a lot of bacteria. And these bacteria do a lot to regulate our immune system. And a good friend of mine, so Bertrand Routy, published some of the first work showing that, I'm just going to show you this, showing that a healthy microbiome is actually required for cancer immunotherapy. So in this experiment, you treat mice with an anti-PD-1 antibody, and you see an anti-tumor effect. But if you treat the mice with an antibiotic that depletes the microbiome, you lose the activity of your immunotherapy. So having a healthy microbiome is important. And it's important for the patient, not just for the mice. Look at this data. Okay, think about it. These are patients that receive immunotherapy. And we're looking at blue at patients that have received an antibiotic before immunotherapy versus patients that did not receive antibiotics. So you can appreciate the difference in survival. It's very important to have a healthy microbiome. And we can exploit that and go a bit further. And what we're doing in Montreal with our colleagues is transplanting fecal extracts from a patient. I know it's disgusting, but it works. So you take fecal samples from a patient and you transplant that to another patient. So my colleague, Bertrand Routy, so he takes bowel preparation from a healthy donor, and he encapsulates those into a pill. The pill has no taste, no discomfort, okay? If you stop thinking, you'll be fine. So the patient takes this pill, and the pill is able to reset the gut microbiome. And he did a worldwide first, so it's a first in the world, first fecal transplant plus immunotherapy in patients with advanced melanoma. And although there's no comparator because it's a single arm study, you can see that most patients had a very striking response. So the percentage of response is higher than what would be expected for this population. So normally it's about 50%. Here we're more in the 70% response. So I hope you liked my talk today, and I hope you were stimulated by it. And don't forget, what is now proven was once only imagined. So imagination is more important than knowledge. That's Einstein that said it, not me. Thank you very much for your attention, and I'll take any questions. Yes.
Radiation therapy is immunogenic cell death inducer. Have you used radiation therapy together with any or anyone in your team with any of these biological therapy to study
Yes.
the synergy, the response?
Yeah, I like your question very much. So also because my field of expertise, the adenosine pathway, gets upregulated in response to radiotherapy. So we've convinced a pharmaceutical company to initiate a clinical trial a few years ago. So the clinical trial was in luminal B breast cancer, where the patient received radiotherapy plus the immunotherapy. Unfortunately for that trial, the addition of the CD73 molecule did not add to the standard PD-1. But the PD-1 versus radiotherapy alone, yes, there was an advantage. So there's an advantage of adding immunotherapy to radiotherapy. Now, it's not a black or white situation because, as you're probably aware, the immunostimulatory effect is also accompanied by some immunodepletion effect. So some cells are depleted right after chemo, but then there's an inflammation that triggers. So it's a complex story, but there's more and more study looking at that combination. You're welcome.
Okay, another question here.
Okay, this is the first time that I met scientists that it's developing a clinical trial, and that's kind of exciting for me. So I really want to know this. What are you think are the main challenges to take the CD73 inhibitors from the clinical trials to a clinical environment?
Yes. Okay. Right now, we're kind of in a situation where the excitement around immunotherapy is waning down a little bit. So a couple of years ago, everyone was super excited. Now we realize that PD-1 is probably the exception. PD-1 was so successful, so important for different cancers. I think the biggest problem at the moment is to identify the patients that will most benefit from this treatment, because it's not a one-size-fits-all. We need to work on the biomarkers. We need to identify which population will most likely benefit. And also because we cannot afford to treat everyone to see only a 10% response. Okay, so that's the challenge. Identify the right population for the right drug.
Other questions? Take advantage. Not every single day we have an expert like John here.
Yep.
John?
There's a question in the crowd. There's a
yeah.
question.
So I know that at a research level it's a little complicated, but what's the estimated cost currently of per patient or per, I don't know, cycle or however it's measured of oleclumab treatment?
So oleclumab is not a drug that is used in the routine practice, so there's no price to it. It's only an experimental drug. But for PD-1 inhibitors in North America, it's around 150,000 per year per patient. But like I was mentioning to many, these antibodies, PD-1, will be off patent in 2 years, or about 2 years. And so there's an opportunity for biosimilars. And there's many companies developing these biosimilars. And hopefully that will reduce a lot the cost.
More questions? John? This lady is watching you.
Yes.
Following you.
Oh, she is.
YouTube.
Oh, hi, Alison.
Alison.
She was in my lab.
She's Costa Rican. Bit of background. She contacted John. She was studying pharmacy here. She contacted John, asking whether he had a place for her to go for a 6-month period to do her TFG. He accepted it, accepted her, and yeah, they know very well. And she was pushing so much for John to come here. And when John came, she got accepted at George Washington University to do a master's on bioinformatics. So now John is here and she's not here. But she is on YouTube, and she sent me a question, John. She's saying, how can CAR T therapy efficacy be impacted by the idiosyncratic difference between patients and varying levels of intratumor heterogeneity?
Yeah, great question. So as CAR T therapies are moving into different types of cancer, such as solid cancer, it's going to be important, I think, to combine CAR T with other modalities. So if you think of a tumor that is rich in adenosine, well, maybe it'll be important to combine the CAR T cell with an adenosine inhibitor, for instance.
Right. I don't know if there are further questions. Otherwise, I'll thank so much, John. It's been an exciting weekend, or exciting week. Exciting weekend.
Tomorrow weekend.
He's waiting for you, the snow, and showing the snow.
Yes.
John has given 3 wonderful talks during all these days. And really, I mean, not every single day we have the opportunity to invite people as John. But more importantly, it's important that university has the opportunity to bring people like John. We have to learn from them. And yeah, that's the only way we can improve our science and encourage you guys, the young guys that are here, there are many, that we need to pursue science. As John did, at some point, he found a molecule, CD73, and he pursued until now that he got a clinical trial and the molecule is doing successfully. That's fantastic stories. We have to dream, because dreaming is the only way we can achieve things. Absolutely.
So thank you for your hospitality. Thank you very much.
Thank you very much. Muchas gracias a todas y a todos. Pues fantástico, ¿no? Tener personas como John por acá. Les agradezco mucho a todos los, bueno, la gente del curso. Recuerden, bueno, al final esto es una iniciativa de un curso que hace 8 años hemos ido empujando, el curso de biología tumoral. y siempre intentamos hacerlo lo mejor posible y para hacerlo lo mejor posible tenemos que invitar gente que nos venga a nutrir. Entonces el curso ha ido creciendo, ya tiene 8 años, ya es un niño empezando a llegar a la adolescencia. Y pues gracias a la iniciativa hemos tenido que, bueno, yo contentísimo porque al final me cuesta un poco organizarlo, pero gracias a la ayuda de mucha gente lo logramos y logramos que la Oficina de Asuntos Internacionales, la universidad nos apoye para traer personas como John. Muy bien, que tengan feliz tarde a todas y a todos, a los que nos siguen en la conferencia en línea y a todos los que están por acá. Gracias, hasta luego.
Thank you for the invitation.